All right, so we're now moving on to the next part of the symposium. So I'd like to introduce now Pablo Meyer Rojas, so who is going to introduce the Allen Cell Lineage Reconstruction Dream Challenge and chair the session as well. So Pablo is a research staff member and team leader at IBM Research, and he's also been the director of Dream Challenges and the chair of the RSG Dream Conference. He's been actively involved in the development and execution of Dream Challenges for a decade now, since 2010, and has led challenges addressing critical questions in systems biology, including parameter estimation and gene networks, and in multiple biomedical fields, including infectious disease as well as developmental biology. And I think we at the Allen Discovery Center have certainly appreciated his leadership in really constructing the vision for this Dream Challenge and executing on it. And we're about to hear some of the outcomes of that today, as well as more about the challenge itself. So Pablo, please take it away.
Thanks, Jay, and thanks for this great meeting until now. I learn every time I come to one of these, and it's in one of these that this idea started about a year and a half ago, and I've learned a lot through this year and a half. I just want to thank all the people that participated. So you and Mike, who spearheaded this, Alejandro Granados from Mike Lab, Max Telford and European Salvador from UCL, and Ehud Shapiro and Ophir Raz from Weizmann. And we kind of all met a year and a half ago, and it's really, I think, through this physical interaction and chat that we came at that meeting, we started putting the ideas together of how we wanted to make a challenge. Finally, I also want to organize the people from Sage Bionetworks that helped putting the setup together to organize the challenge. And so really, as I said, this started about a year and a half ago, and we were going to present the results back in March. And as Mike said, there were a few global changes that made that impossible. But basically, this is just to give you an idea of how long it took us. And actually, it was probably one of the fastest challenges because I think the data sets and the ideas were ready, and there was a lot of willingness to kind of try to push this together. So it took us about a year to wrap everything up. So it is possible to make something like that happen in a very efficient way. The basic idea was, we have all these new tools to reconstruct trees, but we have kind of solutions that have come up from phylogenetic studies, and also new solutions like what Alan presented today. But is there a way to kind of spearhead this and accelerate this process of trying to reconstruct trees? And one of the ideas that came up is, well, why don't we generate many data sets and also give them the solutions to these data sets, meaning how you reconstruct these trees, and see if with that you can generate an algorithm that then could be applied to new data sets where we don't have the actual solutions for the trees, right? So basically just a clear kind of machine learning approach where you give a test set, a training set and a test set, and you try to predict the test sets. And so I think this is something that kind of has been missing in the field, really the need to benchmark the algorithms. And I think you'll see the results are pretty exciting, at least in my view. So the first challenge we took advantage of, and of course the main problem here in cell lineage tracing is we don't have the solutions of the trees, right? There's very little organisms from which we know what really the shape of the tree is. And so we took advantage, and you'll see Alejandro Granados from Mike's lab will talk a little bit more about it. So we took advantage of the fact that they had generated an in vitro data set of mice stem cell dividing under a microscope, and they could actually at the same time read the barcodes to reconstruct the cell lineage, but also image the divisions and have the actual tree. The second challenge that we thought, this is more an in silico challenge, and of course it has to do with the problem that, as I said, there's not many gold standards where we have the actual trees, we decided to go higher in scale, which is something also that I think was important in the ideas around this challenge. So instead of the about 100 cells that we could get from this in vitro data, let's try to do a cell lineage that has 1,000 cells and see how well we can reconstruct it through the generation of in silico barcodes. And of course, the third challenge was going to be something even larger towards the limits of what we have until now generated experimentally, which would be something like 10,000 cells, can reconstruct a tree of 10,000 cells. In this case was in silico, and the idea was to phenocopy a mouse cell lineage, which is probably one of the next frontiers experimentally. So in order to, a lot of the discussions were, how should the trees be, homogeneous, heterogeneous, symmetric, asymmetric. So what we decided to do is just cut through that and actually use the C. elegans tree. Of course, we never told the participants that we're using this tree. And so we masked it a little bit. That's why it's just 1,000 cells and not the 1,000 plus. So the idea would be that we have the tree, but nobody else knows that this is the one we're using. And so this way, it's an in silico data set, but it is a real tree. So we can really kind of try the methods on this. And so the third question we wanted to ask, beyond the fact that can we benchmark, how can we go the larger the trees are, the third question was, can we actually are the training sets useful? And so we generated training sets for each of these three challenges where we would give both the barcodes data and the solutions. And so, for the first challenge, it's pretty obvious. We just divided the data that was produced. For the second challenge, what we did is we used the subparts of the C. elegans tree and ran the algorithm using that, so smaller trees. And for the third challenge, also we used the same EST algorithm that was used to generate the trees, to generate trees that are smaller. And so the idea would be that, not only we know that we can train algorithms, but also that we can use information in smaller trees to try to predict much larger trees, which is something that I think in the future could be important, right? If you want to solve the mouse tree, could you actually first maybe, use an organoid like what Barbara was showing, train your algorithm on that, if you can also use the light sheet to obtain, the tree. And once you have an optimized algorithm for a smaller data set, a smaller tree, can you actually, one of the ideas is, could you use it to a much larger tree? And as you will see in the conclusions, the response is yes. We're happy with the participation. There was a lot of interest, and that happens in general in Dream Challenges. 180 teams participated. At the end, there were 11 teams that participated in several of the challenges and submitted and gave 22 submissions. And we also use different metrics, which is something we use in Dream, but I think in general, people when they're reconstructing trees just use one metric. So you will see also from the presentations that it's important to use several metrics. These are just the results for the first challenge. I just want to show you briefly, Alejandro will go more in detail, but basically, several teams reached a good performance using different algorithms. And also for the C. elegans and the mouse trees, one algorithm was clearly much better. This is declear that Wu-Ming will be presenting a little bit later. So I just want to congratulate the teams that were best performers in these challenges. The first is Wu-Ming's team Kwak-Gong that was best performing in sub-challenge 2 and 3, so the C. elegans and mouse 3. And then in sub-challenge 1, there were 3 teams that were tied, plus Renata that will be presenting today, that I think had a very original approach that at the end ended up doing as well as all the others in a post hoc procedure. And you will see that what's interesting, and she's really using a pure machine learning approach to extract features and to train and reconstruct the trees. So with that, I'll just pass it along to Alejandro. I want to introduce Alejandro. So me and Alejandro actually never met, only virtually, but we share the fact that we both studied at the National University of Mexico, a bachelor's degree. He did then a PhD at the Imperial College in Edinburgh with Peter Swain. Which then joined as a postdoc Mike Allowitz's lab. His main publications and projects are related to cellular signaling, computational biology, information theory, and reconstruction of lineage histories from synthetic recording and single-cell RNA sequencing. So with that, Pablo. I'll just pass the flame to Alejandro.
Thanks, Pablo. Yes, so I think I have control now. Yeah, so my name is Alejandro. I work at Michael Allowitz's lab. As part of the memoir team, I have developed methods for reconstruction of lineages. So it was my pleasure to help with this organization of the DREAM Challenge, and a big shout out to the DREAM Consortium, the organizers, and all the participants. So as Pablo mentioned, sub-challenge 1 was based on the experimental data from INT Memoir, which is a lineage recording technology that has been recently developed at Michael Allowitz's lab. So I'm going to briefly introduce the INT Memoir and the sub-challenge 1 dataset, which consisted of a lineage experiments using INT Memoir stem cells. So INT Memoir is a technology for recording lineage information in situ at the level of individual cells. And this technology was developed by the amazing teamwork of Grace Cho and Mark Booty from Mike's lab. And the dataset consisted, it's now part of a manuscript, which you should definitely check out for more details on the technology, because I'm gonna just introduce it very briefly. So INT Memoir works by the recombination of the action of serine integrases that upon induction create irreversible changes in these synthetic DNA-based recording units, which you can see here. The edits can be read out in situ using single molecule FISH and automated pipeline of image analysis. Then the array states can be used to reconstruct the lineage histories. So as Pablo mentioned, the important thing about this type of technology is that we have access to the ground truth. So we have the time-lapse movies, and we can validate the accuracy of the reconstruction, which is one of the main reasons we decided to use this data set for benchmarking the reconstruction methods. So this is an example of INT Memoir array, which consists of basically 10 recording units that undergo stochastic recording, which is irreversible again. And there are 3 different states. By the way this system is designed, there are 3 possible states. The unedited state is this represented here by number 1, and there are 2 allowed transitions. So basically, before editing, the promoter is expressing the forward Barkov transcript, whereas after recombination, it can express either no transcript, which we call a deletion, or the reverse complement, which is an inversion. And here you can see, basically in this matrix, what we call the digital or discrete states, in which each cell has the 10-unit array, and for each array we can assign accurately which state that recording unit is in. And this is basically a data set that we provided the participants. So matrices in which you have all the cells with a cell ID, and then the 10 recording units and the state for each recording unit. And the full experimental data set consists of like about 100 lineages. They all have, because of the nature of the experiment, they have different sizes and different topologies. So these are not necessarily even lineages with regular shapes. And we can see the distribution of sizes. We have about, the average colony has like 10 cells, and the largest colonies have around 30, 25, 30 cells. And here I'm just showing the frequency edits for the different units. So we can see that it's not homogeneous as you would expect, like 1/3 of each of the states. And this is something actually that the participants use to reconstruct the lineage. So there is information in this uneven frequency of the edits. And basically what we did was to partition this data set between a training set and a test set, as Pablo was mentioning. So we provided 70% of these lineages to the participants with the ground truth, which was again extracted from the time-lapse movies. And then for the other 30%, we just provided the matrix and the barcodes. And of course, the idea was to evaluate that part against the time-lapse movies that the participants didn't have access to. So as Pablo mentioned, we have these two different metrics to evaluate the similarity between trees. There is no really like a consensus on which metric is the best, especially because I guess comparing trees, lineage trees to ground truth in phylogenetics is not like a thing yet. But we decided to use both. And basically the idea is that they consider the partitions of the trees and they compare, for example, in the case of Robinson Falls, from all the possible ways in which you can cut the tree, how many, what is the fraction of partitions that are shared between the ground truth and the reconstruction? And for the case of triplets, the idea is to subsample all the possible subtrees of size 3, and then again count the fraction of triplets that are shared between the reconstruction and the ground truth. And with that, we created this ranking that Pablo just showed you, in which we noticed that the ranking is not the same for when you consider the different metrics. You can see that by the color. So here you can see that the order of the teams is not necessarily the same, although we could see that the best performers were, there was a tie between these three teams, and actually Pablo had to figure out like a way to maybe try to untie this, the winners with a third metric that is not shown here. And also, as Pablo mentioned, this team Amberland, we just want to say an honorary mention because they were allowed to resubmit after the challenge was closed. And they actually matched the top performance as well. So you will hear about that in just a minute. So one of the things that we note as well is that the size of the colonies was one of the main factors to affect the accuracy. And also, as I was mentioning, the two metrics are not necessarily correlated. So we can see here under the two scoring metrics, we have all the teams, and we can see that overall there is some agreement. But for example, one interesting case was the first submission from Amberland. They performed really well under the Robinson Falls distance, which is here. This is the highest score on the lower corner. So they performed really well on the Robinson Falls distance, but not as much on the triplet distance. So which we thought was interesting. And they actually resubmitted their code, an improved version, which is Amberland Star here, in which they actually perform really well in both metrics. So this is why we think it's important to basically understand these two different ways of assessing tree similarity, because they are telling us different things. And the other thing that we noted from the scoring was that the accuracy really decreased with the size of the trees. So in this histogram, again, the lower corner is the high accuracy, and these are the individual trees by the different teams. So you can see that this is the color code for the size of the tree, and all of these large trees from 20 or more cells typically has like lower accuracy. So this has also to do with the fact that the data is not perfect, and there's some amount of redundancy in the recording units. So at some point, there's no more information or not enough information to actually reconstruct these large trees. So yeah, it's pretty exciting that this we have been working with the different teams to basically create documentation for the different methods, and also publication about the Dream Challenge, where we compare the different metrics. And here are basically now they are all available online, and we will try hard to basically make this user-friendly so people can test these different methods with their own data. And you will hear all of this, the explanation of the different methods in just a minute, so you can basically understand which type of method could address better specific questions. And just finally, one of the things that we thought from the beginning with the Dream Challenge was to ask whether a consensus tree combining the different themes would perform better than individual trees. And this was an exciting question to try out, just to see if different people bringing different approaches and different uh, ways of processing the data would outperform the individual teams. And we found out something interesting, which is that the consensus tree actually outperformed all the individual teams, but only when considering the Robinson Falls uh, metric, which we thought was interesting. If we consider triplets metric, then the consensus is not doing as great. And this is both because of the way these two metrics work, but also because of the way the consensus algorithm works. This is a very simple majority rule consensus tree. So one of the things that we would like to do as well is to come up with better ways of integrating reconstructions from different methods, because that could possibly improve the overall reconstruction of consensus trees. And this is another way of looking at this, which is the bootstrap support or the agreement across teams at different depths of the lineage tree. So for example, we pull all the data together and we normalize the depth of the trees. And then we just created 4 beans of depth in the lineage. And we can compute using these 2 different support metrics, the agreement or how well different teams agree that a particular branch is it's happening here. And we can see that towards the end, there is like high agreement and most of the teams can't resolve the sister relationships at the very tip of the tree very well. But basically you can see here that the earlier branches are where the teams disagree. So they would think that the first partition is in this way, but the other team wouldn't necessarily say the same. So there is the conflict here at the very steps. And this is something that is related to why these 2 metrics also are telling us different things from the consensus tree. So yeah, so I just want to thank all the participants and the organizers for the whole Dream Challenge, specifically for Sub Challenge 1. Congratulations to the winners that you will hear about just now. And also thanks to Grace and Mark from Michael's lab for all the support with data and for developing the technology.
Thank you, Alejandro. Thanks for the presentation. There will be option to ask questions after each of the next 3 presenters. Following, we want to show, so as we said, the honorary best performer for Sub-Challenge 1, Renata Rektuke. She's a research associate in epidemiology and modeling group at the Department of Plant Sciences at University of Cambridge. She's working on developing epidemiological models for surveillance and management of infectious disease. Her research has focused on modeling of biological systems at different spatial and temporal scales, DNA replication, photosynthesis, neglected tropical diseases. She's interested in Bayesian inference and machine learning approaches. Renata's talk is entitled A Machine Learning Approach for Cell Lineage Reconstruction. And we're very lucky that she got interested into this challenge to apply her knowledge. Thanks, Renata, and welcome. Looking for your presentation.
Okay, thank you. Such a great meeting, and I had to learn a lot. So I'm a leader of Amberland team, and from the beginning, we were interested in applying machine learning framework. So as Alejandro told about Sub-Challenge 1, it seemed so easy because the data was just 10 targets and 2 mutations. However, when we looked at the training data, it turned out that all cell colonies had sort of different number of cells. And then that meant that the ground truth, the reconstructed trees, had quite different structures. So we have to think hard how to extract what are the features and what is the outcome of our machine learning algorithm. But the main assumption we did, we assumed that we have mutations, which each mutation, each position is independent. So, and the rate of mutations into one or other edit is also independent. So this gave us idea how to get those features. So we decided to look at pairwise comparison. And because we also assumed that it position independent, we thought that we could shuffle all the positions, and as long as the order is in the same colony is preserved, it would be the same ground truth cell reconstruction. So when we looked at, when we decided to look at the pairwise comparison between different cells, we looked if both positions were not edited, if both had the same additions, if one was edited and one unedited, and if they both had different edits. So in this way, we could assign for each position this particular feature. And because we assumed that they are independent, but we could make a new record, which basically just counted how many of positions were both not edited. They had the same edits, and they had one edit, we had different edit. And then we assign status 1 if those cells were sister cells. So there's another example of two cells which are not sister cells. So you can see that this record is slightly different because now we have that 5 of the positions had just one edit. So for our machine learning setup, we aggregated all training data. So it was 76 different cell colonies, and we extracted pairwise features from all cell colonies, which give us about 500 sister cells, records of pair sister cells, and about 3,000 records of non-sister cells. And when we use just this record entries to do machine learning, in particular, we used gradient boosting machine, just because my favorite method. So when we trained our machine learning to get these probabilities, if the sister cells are, if the cells are sisters, we had this output of relative influence. So we found that both the most important feature was that the cells were both the same edits. And the second most important feature was that the cells had one edit. So of course, we tried to look at from the machine learning point of view to run two tests. One was leave one cell colony out and train the machine learning on others. And another is to train everything on 80% randomly chosen cell colonies, and then to test on the rest 20%. And the result, as you can see, those predicted probabilities of sister cells against the observed truth of sisters or not sisters were quite good. But we had some cell colonies which were harder to predict. So we had this idea about this machine learning and getting those probabilities, but of course we needed to do clustering. So from the cell colony records, we could use machine learning to get this probability matrix, which basically this pairwise probability if particular cells are sister cells. However, the question was how to use that to reconstruct lineage tree. And the idea we had is to use thresholds for each level of the tree. So to explain this, I just will use this example. And for now, I assume that we already have this set of non-increasing thresholds. So this is actual 15 cells. And you can see that, for example, bottom 4 had identical records. However, our algorithm doesn't have concept that these are identical. And most probably it never seen this type of combination before. So this is our probability matrix based on the machine learning algorithm model, and each row and column on this matrix corresponds to cell ID. So for example, if you look again at 1, 2, 3, 4, it is the darkest green shade, which means that the probability is almost 1. So now having these probabilities and all pairwise comparisons, we rank all pairs according to this probability. And we start from the highest ranking one. And if that pair is higher than our threshold, we assign that this is our, these are, we cluster them as a sister cells. Then we remove all those cells from another pairs, and then we look at the second highest one and so on, as long as they are higher than this threshold. So basically, based on this matrix, we have that we clustered 7 and 8 as the highest ones, and then 12 and 13 and so on. So overall, we have 8 clusters because we had the 11 one just hanging out on its own. So at the next level, now we look at the pairwise comparisons within those clusters. And then for each cluster, for example, for previously cluster C4 and C5. So for C5, we take a particular cell, and then we pair that with each cell from C4 and looks for the maximum probability from our original probability matrix. And then we do the same with with cell 3, and then look for maximum between 3, 2, and 3, 1. And then we look at the maximum of maximum. So basically for each cluster, we define the highest possible probability between each pair of those clusters. So based on this, now we can cluster old clusters C2 and C3 into new cluster C1. And then we repeat that for next level. And now we will look again all the possible combinations of clusters, and then we look at the all possible combinations between cells in those clusters, and we find then again maximum of maximum of maximum. So in this way, we can sort of reconstruct. But as I said, at this point, I assumed that we had those thresholds. So that is, but it turns out that was a very important part of algorithm. So to find those thresholds for each of the generation, what we did at the end, so basically I did gridded search for the possible thresholds and looked at the training data and what kind of progress on false and triplets distances it would give, and looked for the values for the sequence, which would maximize mean of triplet distances and mean of Robin false. So graph on the left shows overall metrics if I would assume that all thresholds are 0. So basically it would be just strictly binary trees. And on the right is one which is optimized for both metrics. And actually that's paid off. That's how we got this, the best performing entry post competition. However, I should ashamedly say that our original one, for original one, I haven't optimized anything. I just took 0.8 as a threshold for all my generations. That's why the lowest, the test division into sisters worked so well, because usually those probabilities are very high, like 0.9 to 1, but it really messed up the rest of the tree. So fortunately, you haven't heard much about sub-challenge 2, but it was more complicated because it had 200 targets and 30 mutations, and then it had deletions, and mutation rate was in constant for all mutations. It had big range of values. So for this sub-challenge, we sort of decided to use similar concept, but with small changes, where again, we did a pairwise comparison, but now we just counted as a futures numbers of both mutated, both different, both missing, one missing, then both which had mutation A, B, and also we ranked all mutations according to their frequency for each colony, and we then counted most frequent mutation, less second most frequent, and so on. So after training gradient boosting machine for this data set, we had this ranking of features. So what is interesting that the top two are the same as for challenge one. So that, but they switch places. So now having just single mutation was most important, was most relevant in future, and the same all was second. And again, we tried to optimize our thresholds, which was not that successful compared to sub-challenge one. And the outcome, so our results are in light green. So they are not as impressive as for challenge one. So I think it was more like proof of concept. There is still much work to do. And one thing which is missing, I think, with thresholds, I'm still thinking it should be some way to use machine learning to adapt thresholds for each individual cell based on the training data, because because the construction is very sensitive to the values of those thresholds, especially for triplets. And I guess another trick would be to try another machine learning methods, because we use just gradient boosting machines. But I guess that we haven't done that just because we spent 90% of time thinking about how to get features. So thank you for your attention.
Thanks a lot, Renata. I think we're running a little bit late, so we'll try maybe to push the questions for later. But as you see, it's a very original method that Renata developed. So now I want to introduce Wuming Gong, who is an assistant professor at the medical school at the University of Minnesota. Wuming has a broad background in developmental biology, molecular biology, bioinformatics, and computational biology. He was trained in genetics and genomics and has more than 10 years of experience in bioinformatics and computational biology. He is an assistant professor at medical school in Minnesota and focuses mainly on developing algorithms for single-cell RNA-seq visualization and imputing dropouts in single-cell RNA-seq. In addition, Wuming collaborates with other researchers on characterizing novel genes, microRNAs, and long-coding RNAs. He's the best performer of sub-challenge 2 and 3, and as you will see, Wuming was able to impressively adapt existing tools and also generating new ideas to beat the state-of-the-art FAST-3-2 for the C. elegans challenge. So please, Wuming, go ahead.
Morning, everyone. In the next 10 minutes, I will continue to present various algorithms developed for reconstructing lineage tree in this challenge. So I will first give an overview of all methods developed for 3 challenges, and then I will focus on a few distance-based methods for sub-challenge 2 and 3. So let's first define the problem we're facing. So we have a lineage tree, we have a root cell, which state are all zeros, which are the ground states. So each edge represents a cell division. There are internal nodes which state are unknown. The state of all the tips of the tree are known. Each tip represents a single cell. So the input data sets is 1,000 cells, each with 200 Cas9 targets. This is for challenge 2. Each target is represented as one of the following states. The outcome state is from A, B, C till Z, and the ground states, as well as the deletion states. So the ground state must mutate into any outcome state with state-specific mutation rate. The deletion can happen to erase previous mutations. So the mutation and deletions are irreversible. So the whole goal of this challenge is to reconstruct the lineage tree from the input data matrix. So there are 28 teams registered for the challenge, and 7 teams made the final submission to at least one of the challenge. And each method employs a different method. For example, the Cassiopeia use a maximum parsimony-based strategy. Amberland and RNA labs use feature extraction followed by gradient boosting and hierarchical clustering. So on the other hand, the majority of this method, especially for challenge 2 and 3, are distance-based, which first estimate the distance between the sequences and then reconstruct the lineage tree. These algorithms are generally scalable to large data sets with thousands of cells and targets. So the Hamming distance is the simplest way to evaluate distance between two character sequences by simply counting the number of pairwise differences. For example, the Hamming distance for AAT CGC and AAT GCC is just 2. So the problem of regular Hamming distance for these specific problems are each character is equally weighted, while the outcome probability for each state are actually different. And the second reason is that deletion result in large differences, which is unfair for counting the distance. Three groups developed method for address the problems in the regular Hamming distance. So for declare, we developed a simple strategy to assign different weight to each state. The method is called weighted Hamming distance. So it's implemented in an R package called declare. So we optimize the state weight using the Bayesian hyperparameter optimization based on the training data and apply to the same weight to the leaderboard data sets and final data sets. So in Liu's method, the transition probability between different states is calculated by counting the frequency of every state transition from parent node to child node. In the training data, the distance matrix is built using a similar Hamming distance weighting protocol based on the transition probability. The tree is constructed by the UPGMA algorithm. So in Guang's method, the first step, all gap mutations are remarked based on mutation types at both ends, and they generate the mutation weight for each state at each of targets. And the weight is defined based on how important the mutation incident should be considered while comparing the mutation state between cells. The tree is constructed by an iterative clustering process. So in declare package, we also developed another distance-based method, which did not require any training data. So given input sequences, so we first summarize the mutation rate, outcome probability, number of targets, and number of tips. And with some other predefined hyperparameters, we simulate the lineage trees. So, and then we summarize the K-mer replacement distance matrix, which is the nodal distance between every pair of K-mers. So, and then we compute the cell distance based on the K-mer replacement distance. And with this cell distance, we are able to construct a tree based on according to algorithms such as FSME or NJ. So in the following few slides, I will introduce how to derive the nodal distance between the entire target sequences from the expected nodal distance between single states and the K-mer states. So we first show how to use the simulation to estimate the expected nodal distance between single states. So we simulate 1,000 lineage trees with the following these parameters. And in each lineage tree, we randomly sample 100 nodes. For every pair of nodes, we recorded the relationship between their nodal distance and the 1-mer replacement at each target. So as expected, this histogram shows the mean nodal distance between CC is smaller than AA because it's very likely, it's much less likely to have a mutation from the ground state to state C compared with state A. So at the right side, this heat map shows the expected nodal distance between deletion states, ground states, and the few frequent outcome states. So here the X is a single state in sequence I, and the Y represents a single state in sequence J. So D is the observed nodal distance between node I and J in the simulated lineage tree. So here are a few observations here. So first, the rare outcome state is associated with smaller nodal distance. So the nodal distance between the ground state and any outcome state is constant. And the nodal distance between the deletion state with any outcome state is also constant. So this result suggests that one-mer replacement distance is associated with nodal distance in the simulated lineage tree. In next step, we extended the one-mer replacement to k-mer replacement. We found that the nodal distance of k-mer replacement can be estimated from a bunch of two-mer replacement. So that's defined two k-mer sequence, k-mer x and y, where each represents k single state. So simplest way to estimate k-mer replacement distance is by through an independent model, which assumes that every individual state is independent. So another way is actually consider a neighboring state, resulting in a conditional model. So the two histogram here shows that observed states observed nodal distance between C dash and CC, as well as the quadrant B. So the observation here is that distance estimated by conditional model is much more closer to the observed distance. So the independent model, however, tend to underestimate the nodal distance between the rare and unchanged k-mers. You can see here. So the results suggest that nodal distance of k-mer can be estimated from two-mer replacement by a conditional model. So in the final state, with the k-mer replacement distance, we are able to estimate the nodal distance between two sequences. So in this case, each sequence have n k-mers. So we estimate the expected nodal distance between two sequences by simply averaging the expected distance of all pairs of k-mers here. So we assume that here each k-mers are independent. So we use the simulated data to estimate three things. So one is the marginal distribution of the nodal distance. The second is the conditional probability of a single state replacement. And the third is the conditional transition probability. So this new CRISPR-based molecular tool allows us to solve the lineage of complex model organism at single cell level. So this dream challenge is the first attempt to rigorously examine the performance of robustness of various algorithms across many parameters. So several tree metrics are necessary to comprehensively evaluate the performance of the lineage reconstruction. And so the distance method here is popular due to the simplicity and the scalability. So once the distance matrix is obtained, there are many tree construction methods that can be used. And we found that some tree arrangement operations may be necessary to have a good performance. So we hope that challenge will encourage development of more sophisticated and powerful tool for this domain. So here, I thank all the challenge organizer, data contributors, and all the participants. So thanks very much for your attention.
Thanks a lot, Wuming. Once again, we're running a little bit late, so I'll just, we'll introduce Matt, and then at the end we can take questions if there are about all the different approaches. So Matt Jones is a PhD candidate at the University of California in San Francisco and University of California in Berkeley, working in Jonathan Weissman's and Yosef's labs. He holds a degree in computer science from UC Berkeley. Currently, his work focuses on developing algorithms for single-cell lineage tracing technologies and using these approaches to learn new biology. Previously, he has worked on developing computational tools for analyzing high-dimensional single-cell RNA-seq assays and simulating complex population genetic events. He is currently a UCSF Discovery Fellow and was a research intern at Google Health. Matt, please tell us about your approach, your different approach for solving these challenges. Thanks.
Yeah, well, thank you so much for the introduction, Pablo, and thank you to all the organizers, the Dream Challenge, as well as for the symposium. It's been a fantastic event, and I'm very happy to be participating. So I'm representing the Yosef Lab team, which is a large team made up of postdocs, undergraduates, and graduate students, as we were one of the winning contestants for Sub-Challenge 1. We also made submissions to Sub-Challenge 2 and 3, so I'll also be telling you about our algorithms for addressing larger scale phylogenies. But before I get there, I wanted to just begin with what's obviously an understatement considering the talks earlier today, in that reconstructing these unobserved phylogenies is an extremely difficult task. And generally, a lot of people who are a lot smarter than me and older than me have been thinking about this ever since Darwin first noticed that natural selection and phylogenies underpinned all biological variation. And so what we come to appreciate is that phylogenetic inference is NP hard, and generally there are very few regimes where an efficient algorithm can be applied with meaningful guarantees. And in single cell lineage tracing, this is particularly difficult because largely we're dealing with extremely large data sets where this number of cells largely outweighs the number of features. So these are undetermined data sets with quite a lot of missing data, which can confound a lot of analysis, either learning what's important features or just determining which cells are really related to another, or just confounded by missing data. We also have this regime or phenomenon called homoplasy, where the same mutation can appear at different parts of the tree independently and artificially make two cells look like they're related to another, but actually are from different parts of the tree completely. Also, we're imperfectly sampling the cell populations. So we're not sampling every single cell, and this also makes the problem pretty difficult. And like I said, a lot of people have thought about this problem, and theoretically, in small enough input sizes, we can use an approach known as Steiner trees to solve the maximum parsimony problem to optimality. And Steiner trees are a very popular theoretical computer science problem used from circuit design to network layout, and they all operate on graphs. So essentially, one of the inputs for Steiner trees is a graph where you have nodes connected to one another by edges. And if you specify a set of terminals, highlighted in red here, a Steiner tree is the minimal weight tree over the graph, connecting only the terminals. So you don't need to use every single vertex. And theoretically, you can use Steiner trees to model phylogenetic processes. So in this case, terminals are observed samples. So these are our cells that we're interested in reconstructing lineages for. Vertices highlighted in black here are unobserved intermediates, so the tops of the trees that we don't observe. And edges are legal phylogenetic changes. And so, like I said, for small enough data sets, we can apply this approach. And we had to do a fair bit of engineering to make this approach amenable to the lineage tracing design constraints. But essentially what we can do is take our set of observations, which we normally summarize in this character matrix here, where the rows are cells and the columns are target sites or recorder sites, and individual colors here represent states. We can take our observed cells and infer the sets of ancestral unobserved states that could possibly have occurred on the path from the root to the observed sample of cells. Importantly, we had to incorporate how to deal with missing data, how to deal with the fact that mutations are irreversible, and also we had to condition the size of the ancestral state's potential graph by the observed data, because this can just explode in complexity if you're not careful. But then once we have this potential graph, we can then use integer linear programming, which is a very popular combinatorial optimization technique to solve for the Steiner tree. And this is our maximum parsimony tree. There's one important thing that we incorporate here potentially, and that is the edge weights. So we know that certain states are more common than others, especially with Cas9 based lineage tracers, where certain Cas9 based outcomes after the double strand break are much more frequent than others. And so essentially we want to bake these priors into the actual phylogenetic inference. So this is generally how Cassiopeia ILP works. When we were approached the Sub-Challenge 1 data, we had this rare opportunity to learn from the data, from the training sets. And in particular, what we could do is learn the relative propensity of a state going to the deletion state or a state going to the inversion state. And a priori, you might assume that these are equally likely, but actually we found from the training data that there's a slight preference towards the deletion state than the inversion state. We can then bake these probabilities into our Steiner tree inference procedure. And on the training set, if we compare the performance of the algorithm that used the weights or actually baked in the priors versus the algorithm that did not use the weights, we see by and large they agree, but the weights actually do help us out in certain rare scenarios. And this really allows us to ask questions about how the lineage tracing really behaves, or how cell development actually behaves in a ditch where we actually have the benefit of watching these cells divide, which is extremely rare. And I think that our team has learned a lot from this data set. So like I said, the IOP algorithm is not very scalable. For sub-challenge 2 and sub-challenge 3, we applied a greedy algorithm, which is predicated on this heuristic derived from perfect phylogeny. Perfect phylogeny says there's a very special regime. If mutations happen once and only once, you have a very easy algorithm that's efficient that can solve this tree perfectly. And so we can basically use this idea to split our cells into groups that are mutually exclusive based on the presence or absence of mutations that we think happen earliest and perhaps only once in the phylogeny. And we can do this by using a combination of the frequency at which we observe a state as well as the prior probability of that state. So ideally, we'd like to select for states that occur many, many times in our observed samples, but have very low probability, so likely occurred only once. And this will be used for sub-challenge 2 and 3. We see pretty good performance in sub-challenge 2, but unfortunately not too well on sub-challenge 3. And so really we're kind of drawn to understanding what are the trade-offs between these algorithms and how can we improve Cassiopeia greedy. And in particular, there's this trade-off between theoretical accuracy and efficiency. So on one hand, Cassiopeia greedy is very efficient because it uses this heuristic approach, but only has theoretical guarantees if homoplacy or conversion evolution is minimal. On the other hand, Cassiopeia IOP is theoretically robust, but not very scalable. And so we had this hypothesis that if you could avoid homoplacy, you might improve your Cassiopeia greedy performance. And so on simulations where we could actually simulate regimes where homoplacy is quite rampant, such as when you have very few states, you can see that IOP outperforms significantly greedy. But as you decrease homoplacy, for instance, by increasing the number of possible state outcomes from a Cas9 cut, you're able to see relatively similar performances between IOP and greedy. Similarly, if you can tune the mutation rates well to your system or your experimental question, you can also reduce homoplacy. But if you have too high of a mutation rate, such as the regime shown on the far right, you can also see that Cassiopeia greedy's performance starts to tank as well. So really, the most interesting thing you can do, though, is actually see how this performs in real lineages. So we have this hypothesis that homoplacy might be making greedy perform worse or better, but actually in memoir data, where you have very few states, greedy actually seems to perform fairly similar to IOP. And you might ask, why is that the case? If greedy doesn't have these same theoretical underpinnings, why is it performing so well as IOP? And I think one of the reasons is that there's no missing data here. So it's just good engineering. And there's also very low homoplacy, probably because there's well-balanced mutation rates and shorter experimental timescales. So this just shows how good engineering could potentially make the phylogenetic inference problem a little bit easier. And you can use heuristic-based solutions to solve these trees better. So I described the Cassiopeia greedy and IOP. These are two modules in a larger set of tools that we developed, all under this umbrella of Cassiopeia and published in Genome Biology earlier this year. These all are maximum parsimony-based methods, and we're trying to make them as flexible as possible to the complexity of the data and bake in as much prior information as possible. And this is really just a snapshot in time. We think that we can do a lot better, and there's a couple of challenges that we are particularly interested in solving. The first one is developing better experiments that are better algorithms at scale. And we just recently described a set of experiments where we tracked clones of size 10,000 cells or greater. We think that as everything in single cell, the scale is going to just keep on going and going and going. And so the deficit algorithmically needs to shrink, in my opinion. We're working on this. We're also starting to appreciate that missing data can be really confounding to the inference, particularly because missing data can come from two sources. It can either be heritable, coming from epigenetic silencing or large Cas9 resections that get propagated down the tree, or it could be stochastic, which is really something like random noise introduced by the sensitivity of the sequencing platform. And lastly, we're really interested in inferring branch lengths and relative timings on the phylogenies. And this is really crucial, I think, for deriving better phylogenetic summary statistics and understanding the actual biology. But it's tough because a lot of these recording technologies use stochastic cutting procedures. And so you can't use this molecular clock hypothesis to time branches that a lot of people use in real DNA sequences, phylogenies. And as I said a couple of times already, I think that both these problems can be addressed with either improved algorithms or improved technologies. And I think this dream challenge really highlights how different engineering regimes might make the problem harder or easier. And I think highlights the intersection between these two approaches. And with that, I want to thank everyone involved on the Weissman Lab and the Yosef Lab and at UCSF and Berkeley, especially the undergrads who spent way too much of their precious time in undergrad working with us. So yeah, with that, thank you so much. And I'm happy to take any questions.
Thank you, Matt. And thanks, Wuming, Renata, and Alejandro for doing a great job trying to explain a little bit, what came out of these challenges and the importance of having being able to benchmark algorithms in the same data set. And for that, we thank Catherine and the Allen Foundation for pushing this forward and helping us bring this to presenting the results today. So I think we have time for a couple of questions. I think Renata had a question about, there was a question about what do you do with cells that have the same barcode. So if you want to answer that one.
So answers is sort of random, but actually I just trust algorithm, how it ranks, and it most probably the same cells had, different cells had the same records. Every time I run algorithm, my machine learning cell lineage reconstruction, it will be slightly different, but I don't control that. So basically I leave that to algorithm, the way it ranked, so that will be, different cells will be assigned together. But is it strictly binary or just single cell? So I don't allow that it will be 3 branches out of 1 node or 4. Or
Right, and I have to say, we, of course, there's spatial information, but we didn't give, or participants didn't use of the spatial information that could be extracted from the microscope, which is something that was discussed by Cole and also by Alan, I think. Okay, so there are a couple of questions for Wuming. So somebody, Wuming, is asking whether you simulated 1,000 lineage trees and if you use Monte Carlo for that. That's the first question. And the second question is, how do you think your algorithm would perform if you wouldn't have any training data related to the mutations?
So the first question, yes, we are trying to estimate some of the summary statistics from the simulated data. It's kind of Monte Carlo. So we simulate trees, and from the tree we summarize some of the key numbers to estimate the camera replacement distance matrix. So the second question. So yeah, it's a good idea to have for example, the lineage specific mutation rates, but so far our method assume a constant mutation rate for each state through the whole data set. But that would be a good idea to estimate maybe lineage specific mutation rates.
Thanks, Wuming. There's a question from Sophie Cito for Matt. So Matt, how do you plan to infer branch length? Do you plan to extend the Steiner algorithm, or do you plan to use other algorithms?
Yeah, that's a great question. And like I said, I think really important to the future of how to apply these technologies and algorithms to real data. I should first say that there was a fantastic method published out of UW earlier this year, I want to say, from the Matson group that tried to develop a new likelihood model for how Cas9 would be cutting Gestalt targets and show that they could do a pretty good job of estimating the split times of certain phylogenetic splits. But just generally speaking, we're thinking about penalized likelihood models such that we think that branches located in the same lineage should hopefully be similar timings. So essentially, cells that are in the same lineage should be the same age, as well as using some Bayesian methods where the actual stochastic cutting rate can change on a branch-to-branch basis. John Helsenbeck's group out of UC Berkeley has done some of the stuff for Bayesian inference of mutation rates at the same time as phylogenetic inference for real evolutionary biology. We're hoping we can take some of these ideas to the single cell synthetic lineage tracing approaches.
Okay, I think maybe time for one last question. Matt, is also for you. If the end nodes, if the end nodes activate without the whole matrix, what mechanism controls it? Or is it a random activity? I'm not sure I understand the question.
Yeah, I'm sorry, I don't, are you asking about the mutation rate at the very end? I'm sorry, a clarification.
Victor, if you could clarify your question. Okay, not sure. Okay, anyway, I mean, I think we can let it for the next time, or you can replay it on the chat. So I think I'll just pass it on to Jay Shendure for closing remarks. Thanks everybody, and this was great. Thanks for spending this time with us.
So thank you, Pablo, and thank you, Alejandro and Renata and Wuming and Matt for those terrific talks and for your excellent work on the Dream Challenge and sharing the results. Just a few closing comments, and then we'll wrap up amazingly on time. So just again, another round of thanks to our three presenters this morning and those really fantastic talks in the first half, Barbara, Alon, and Cole. Really appreciate your sharing your work, and I know a lot of it unpublished. And I'd like to say again, I know Michael had some of this in his introductory remarks, but this was a lot of work to pull together. So I do really want to thank again the folks who made this event possible. So of course, the whole Paul G. Allen Frontiers group team, in particular, Ashley Bachman, who did a ton of work here, as well as Kathy Richmond, Alexandra Basford, Casey Elkins, Nicole Huber, the Allen Institute Communications team, and of course, the late Allen Institute founder, Paul Allen. I'd like to also extend another thanks to Tish Maskell and Joe Leonardo from our teams for their work in helping with this. And thank you all, the audience, for your engaging questions throughout the day. I know we had a lot of attendance from other invited speakers from the Hindsight 2020 series, as well as folks who would have been there with abstracts and poster presenters and all that. So of course, we remain bummed that we couldn't do this in person, but I think excited that we were able to pull it off in this virtual format to have such an engaging morning. So just one more plug before I wrap up here for the point that Michael made at the beginning, which is that this is going to turn into a series. And I think Ashley will be posting, or hopefully has already posted, yeah, there it is, the link in the chat to our Allen Discovery Center for Cell Lineage Tracing website, where you can register for the ongoing series. And again, just really an amazing set of speakers that we have coming up over the coming year. And then, hopefully this, maybe in a year or two, we'll come together again, once we're post-vaccine and post-pandemic and actually do this in person. So as a reminder also, the recording of the session will be emailed to all registrants and posted at the relevant websites, as well as on the Allen Institute YouTube channel. And finally, thanks again to all of you for coming and for sharing your science and being a part of this, and enjoy the rest of your day or your evening.